Thank you, Jason, and thank you to the university for the opportunity to speak with the group here. So as Jason mentioned, my name is Scott Marty. I'm a partner with Ballard Spahr. I am a patent attorney, PhD in biochemistry and molecular biology, and have had the privilege to work with the University of Utah for over 2 decades now with the Technology Commercialization Office, now the Technology Licensing Office. So really appreciate the turnout. It's been absolutely amazing to watch the growth here in Salt Lake City, to see the group grow, to see innovation grow, to see the community grow, to see funding grow. For those of you, and I know some of you have been around for a long time, you could probably echo these thoughts. So find some of the folks that have been here for a long time, and you'll hear how lucky you are if you're new to this area to be part of this ecosystem. So today we're going to focus on diagnostics. I have several questions for our speakers, but now that I've introduced myself, I'm going to let the team here introduce themselves, and then we'll get right into the content.
Sure. Can you hear me? Well, yes. Hi, my name is Tracy George. I'm a physician scientist in the Department of Pathology here at the U. But my day job is I'm President of Innovation and Chief Scientific Officer at ARUP. I came out of academic medicine. I was at Stanford for about a decade as a faculty member, and then went to University of New Mexico and was finally enticed to come to ARUP and University of Utah about 8 years ago. And it's been a fantastic and wild ride. So it's really nice to meet all of you. Looking forward to some more conversations.
It's a pleasure to be here. I want to thank the organizers for the opportunity. My name is Matt Rondina. I'm a professor of hematology and oncology at the University of Utah. I also direct our molecular medicine program, which is a translational research unit with about 30 labs. My own lab focuses on using sequencing technologies and other approaches to examine and understand gene expression changes in circulating blood cells and stem cells. I have a secondary appointment at the VA Medical Center where I see patients and direct our thrombosis service.
Great. So as you can see, we've got some extremely talented and experienced folks here, and so I'm going to jump right into some of the broader topics that we have to discuss, and you have a microphone to share. So Matt, you're holding the microphone first, so I'm going to ask you this question, and you can pass it over to Tracy. But I want to underscore something. What Tracy shared with you, Stanford, New Mexico, University of Utah, these are all top-tier commercialization entities. So I'm going to kind of preview, which I didn't do before, is to say, I think that the group here might benefit from understanding some of your experiences of places you came from and where you came to when you answer some of these questions, because each of these institutions do things a little bit differently, but they do them very well. So my first question, Matt, for you is, what are the biggest opportunities and challenges in translating academic discoveries into market-ready diagnostics?
Yeah, thank you, Scott. So I think there's several challenges as a faculty inventor in developing technology that's really foundational to some of the discoveries we made almost 5 years ago now. I think probably one of the big challenges is knowing what you don't know and knowing how to fill those gaps. And as Scott said, I think we're very fortunate right now to be in the biotech ecosystem here in Salt Lake City, where we have amazing resources and people available to help us fill those gaps. And so fortunately, I was able to connect with a number of really experienced business folks through the TLO that helped me really understand early on where our own technology could fit a market need, and really how to plan a strategy going forward to, as quickly as possible and as successfully as possible, move that technology down the commercialization path.
Yeah, so this is a hard question because there's a lot of answers to it. So from ARUP's perspective, we're a national clinical reference lab. We're the largest nonprofit reference lab in the United States. We do a lot of business. And when I came here, what I saw is a very robust research and development group that was mostly development. And the research had shrunk over the years. So a couple of years ago, we started a new institute, Research and Innovation Institute. And why, you might ask? Because when someone like here in the audience brings us an idea, it's going to be at different stages. And so for those folks who are more at the research stage, my group at the R&I is going to interact with you. So we're looking at like new tests, new technology 5 or 10 years out, right? Things that aren't really quite ready for commercialization. And then there's folks who've been at it for a while. And then we're looking at taking your research assay, which you think is a fabulous test, right? And you know it's the best test out there, but it's not going to work in a commercial lab, right? It's not hardened, it's not to scale, and that's what my development teams do, is they take your fantastic technology, they make it so that any med tech can run it, okay? And that's super important. And then there's the whole piece of clinical efficacy. So I've had some companies come to us and they say, we've got the best lab tests, it's, PB&J, right? This is fantastic, it does all this stuff. But there's no clinical efficacy, right? It's not been used in any clinical trials yet. It's maybe, not even used in a hospital yet. It's being used in a handful of clinics. So having clinical efficacy is really important. It's also very expensive trying to get your test out there or your technology being used in some sort of clinical trials. So that's why we created our R&I Institute, to interact with folks who are kind of early to this stage so we can help them, right? Help them develop their tests, help them develop their technology in our ecosystem because it's going to be probably 5 years before it's ready for the development team and taking it there. So there's different stages. So I see it as really a ton of opportunities. I'm really amazed by the people that I've met in Utah and the ideas that they have and the willingness to try. The perseverance is pretty amazing, and that people are willing to collaborate and they're willing to think big and and to challenge assumptions, right? Like, hey, why don't we do things so differently, which is great. And but having a good idea is not enough. There's a lot of steps to taking things through the whole life sciences pathway. And luckily, there's a lot of really smart people here in the room and in our ecosystem who can help and advise you about IP, right? You want to do that as soon as possible. You want us to be talking to TLO right away. So those are some answers.
Well, that took away 2 of my other questions. but I want to follow up on that, and I want to get your perspective, and then Matt, I want to get yours, because what I think people sometimes don't appreciate is having an asset like ARUP basically on campus. Like having a commercialization destination for their technologies and being willing to look at technologies at that early, mid, and late stage. So if we focus on the early stage, right? So we're thinking about faculty that have that miraculous idea, they have their grants, they have that funding, they have that proof of principle. When your team is looking at that technology, what is it that they're actually looking at and say, we're willing to invest in helping that investigator move that to mid and then late stage? So you mentioned IP, so IP position maybe is one, but what else are you looking for at that stage in early development?
So, I would say faculty generally share our mission, but whoever we talk to have to share our mission. So you have to be aligned, right? Our mission is taking care of patients, improving diagnostic medicine for folks here in Utah and around the world. So that's very important. And I talked to plenty of people, that is not their mission. Their mission is making money. That's great, but that's not going to align with my mission. And our mission also is, we're not grabby about the IP, right? We want to help you publicize whatever you're doing. We want you to present, we want presentations, we want to be on academic publications together. That's good for my faculty, that's good for your faculty. So can we collaborate with you? That's super important, the people factor. So honestly, those are the 2 probably most important things. Mission, and can I actually work with you? Can my teams work with you? And then we get to, is it a good idea? Is it really going to solve a problem in the market? Or is it a solution looking for a problem? Right? I think we've all seen those, right? So we're looking for problems, and then we want you to have some solutions to that actual problem. And I see a lot of things that require a lot of changes in our ecosystem and health system that it's just not gonna work, right? So, and then, or is it disruptive technology? Or something that really accelerates healthcare? Okay, you know, some of the AI solutions can really do that. And, you know, you hear plenty, well, we can save you, you know, this many millions of dollars. Do you have documentation of that? If you do, that's fantastic. Is it direct that you're saving, or is it indirect? These are some of the things that we're looking at. And, but, you know, I'll be honest, I talk to everybody. People reach out on LinkedIn, however they get my email, I'm talking to everyone because you never know who's gonna have a good idea, right, that you're gonna wanna work with. And sometimes we're not the right partner, but we know of people who are the right partners, right? And we just want you to succeed. So, you know, if we're not the right partner, we're gonna introduce you to someone who maybe is the right partner.
Perfect. So Matt, from a different perspective, as the inventor and innovator of technology, as you're trying to get your technology out there, what do you see as being the major obstacles that you're faced with if ARUP is not your home and you're looking for other homes? Maybe you can talk a little bit about what those obstacles are and what that process looks like.
Sure. So, you know, early on when we started down this pathway, I realized that I needed to have conversations with people who were in the laboratory diagnostic space, understand all phases of the pathway, not only basic things like how do you get to a prototype test, how do you get through basic analytical validation, how do you start to move that out into clinical trial settings where now you have different labs handling the specimens, you need to think about things like transportation and stability and reproducibility. And so we were very fortunate to have ARUP in our backyard where we could have these conversations and very easily engage with people like Tracy and her team members that had been working in this space for a long time and really lean on their expertise to help us understand facets of this. I can tell you in parallel, you know, we had conversations with other laboratory partners that were not as closely connected to the University of Utah, and those conversations were much different. They were much less collaborative and more for kind of fee-for-service oriented, which still can be very valuable. But I think for me as a faculty inventor, having these close connections to ARUP, having this ability to really align on a collaborative project, get early feedback, and then implement that feedback into our developmental pathway was really, really key for us.
That's great. And for those that don't know, if you're in the diagnostic space, if you take nothing else from what was shared so far, there is a direct connection between ARUP and the TLO. It is a bridge that is used often, that is used early, and one that anyone in the diagnostic space, I would highly encourage you to engage in one or the other, because it is, if nothing else, there's databases, there's people power, there's opportunities to take advantage of in this space. So, you know, we've kind of touched on this a little bit, and Matt, while you still have the microphone, you know, thinking about, because you've gone through the process, you know, if you were looking at, you know, new faculty or graduate students who are now innovating these new technologies, what advice would you give them on how to engage and how to leverage the resources here at the university?
I think there's a couple of important aspects. First is engage early and engage broadly. I think, Tracy, you touched on this. One of the really unique aspects of Salt Lake City and the University of Utah ecosystem and the greater ecosystem in general is that I think we're highly collaborative. I think that we're much less siloed. We don't have many of the same walls as other communities have. And that, I think, really engenders early conversations where you can quickly get to know people in the community. I think those people are not only talented, they have deep expertise, but they're really committed to helping you succeed. So number one, engage early, engage broadly, and then realize that the University of Utah, through the TLO and other programs, they have a number of different opportunities where you can not only work through the University of Utah ecosystem to pull in some of those resources you need, but they'll also help introduce you to people out in the community for additional partnerships and expertise.
So, Tracy, I have a different question for you, which is, because you're in the business of commercializing these technologies, what is it that you see as being the major obstacles in getting that innovation through commercialization to the point where it becomes something worth putting the time, money, and effort into launching a commercial product.
Yeah, I think I mentioned this earlier, but it's clinical efficacy. You have to show that your test does what you want it to do and that it's being used by folks in the field, and that's a hard hurdle to get through. You know, some of the easier ways are, for example, if you hook onto a clinical trial as an exploratory endpoint and you're doing the testing. I mean, that's one way to do it, right? That's probably the hardest part. So folks do think that they have a great lab test and that we will just put it on our menu. Have you talked to LabCorp and Quest? Do you know how much it costs to put it on their menu? Right? I mean, you know, it's thousands of dollars that they, you know, there's an exchange here. We don't do that, but we only put tests on that have clinical efficacy. So the bar is high. It has to have been proven, and we understand most people haven't got there yet. So that's why we introduce you to folks and try and help you develop the tests enough that it can be, or the technology, so that it can be started to use in some way in clinical practice, so that you can get papers out there that show that your test is useful, for example, in prostate cancer, right? It's like, well, maybe you're gonna start in Europe first, and then you're gonna go to the US, right? Or maybe you're gonna start somewhere here and then go somewhere there. But you have to start somewhere where you have traction. And I you know, so it's really, that's the biggest stumbling block that I've seen.
So are there, I'm gonna ask you, and then Matt, I'm gonna ask you, because you're starting a technology and sometimes choosing what particular indication you want to pursue first, right? Because, you know, unfortunately, we're not full of cash, right? As an academic, we have to get funding. As a company, we have to get funding. As ARUP, we have to get funding. And we have to find ways that we want to spend our money. Are there particular indications or areas that you're seeing that are currently hot in diagnostics that if you were to put like your top 3 areas for diagnostics, what fields might those be that you would be looking for now?
So you might kill me, Matt, but I'm going to focus on non-neoplastic diseases. I think oncology in some ways has been tapped out, not completely, but like, you know, the neurodegenerative space, oh my God, that is super hot, right? Number 1 spend in this country is what? Neurodegenerative disease? Okay, you've got to go there. And then there's the inflammatory disorders, right? Those biologics, hugely expensive. I mean, so one way to look at this is, you know, you look at where is pharma developing new drugs, right? We just started developing drugs for Alzheimer's disease for patients with symptomatic disease, and now we're moving into pre-symptomatic, and then there's, of course, a whole variety of different neurodegenerative disorders, of which I am not an expert. But then next, I really think the inflammatory disorders are really going to be where it's at. There's a lot of biologics that are being developed for things like, you know, rheumatoid arthritis, other inflammatory disorders are very hot right now. I mean, all the drug manufacturers, right, that are making the GLP-1s, we've all heard of that, you know. I won't ask how many of you are microdosing out there. But you know, but we all know, right? They have anti-inflammatory effects, right? So they're going to be taking those drugs, and they're trying to right now apply them to other disorders. So those are like number 1, number 2, and there's a lot of different inflammatory disorders. So, you know, I think that could maybe trickle on.
So Matt, as someone who's, you know, looking at new codes and fundraising, as you're talking to investors, you know, what are the areas that you're hearing that they're most interested in?
Yeah, thanks, Scott. So I actually agree with you, Tracy. We've thought a lot about this, and while oncology is our lead program for reasons I can get into afterwards, we've looked carefully at both opportunities in the neurodegenerative and inflammatory diseases, mostly in some of the inflammatory arthropathies,
Yes.
which are difficult to diagnose, which can be incredibly disabling, and where now that there are advances in treatments, early accurate diagnoses can really prevent a lot of the disabling sequelae that can occur in those patients. So I think those are exciting opportunities as well. And just to touch on something you said, Scott, as well as you, Tracy, we've thought a lot about clinical efficacy and really from early stages, thought carefully about where our intended for use populations could be, where we could most feasibly demonstrate clear clinical impact, either by improving care, whether that's early detection, less invasive detection, or whether we can help providers more rapidly and efficiently assess people that have true disease, meaning they actually have the condition, versus patients where they have other conditions that should be evaluated and treated differently. And one of the ancillary projects there is actually figuring out how we can get these tests into the hands of patients in less accessible regions of the world. So not only thinking about major academic medical centers where we do have very sophisticated diagnostic medicine available, but how can we actually develop these into a test that could be deployed in more inaccessible, geographically isolated regions where we know many patients don't have access to the same level of medical care, whether that's diagnostics, whether that's treatments, whether that's access to specialists. We're really trying to improve accessibility to these tests as well.
How are we on time? We're good? Okay, great. Just I want to make sure I reserve, because we're not taking questions during the meeting, but I want to make sure people have an opportunity to speak with Tracy and Matt afterwards. But I can echo, I mean, as a patent attorney, I see a lot of innovations, hopefully if they're doing it right before anyone else in the world does, so there's no public disclosure. And those areas are areas on the therapeutic side I see as being very active. Something else that I've seen as a trend in what I do is the difference between diagnostics, meaning you currently have the disease, and the diagnostics of you have a propensity to develop the disease. And so all diagnostics, but also very different markets, right? Because if I'm diagnosing a disease, then I'm telling you have this disease, and I'm looking down my checklist of the different therapies I can use. If I'm looking at it from the context of a propensity to develop a disease, what are you seeing in that market? Like, are you seeing more innovation coming there? Is that something that ARUP is looking into? Or is it more of, we want diagnostics that are looking at ways to then treat a disease versus how to prevent a disease?
It's interesting. I think our healthcare system does a better job with treating already known disease than trying to do preventive medicine, even though we all want to get there. We're of course very interested, right? It's like we'd like to treat your Alzheimer's disease before you develop it, so we can slow that down rather than after you develop it, right? That's an obvious one. There's already boutique labs that are testing patients who show no signs of Alzheimer's to detect those who have a higher propensity to get there. But there is a debate about that, because the debate is, how much can you actually do? Or for things like Huntington's, for example, right? These are hard things. But you're really getting into also the wellness space. And so that's what we're seeing a huge growth in. I think this is direct-to-consumer testing in wellness, right? Hims, hers, rhythm, I mean, you name it, right? But do you know that like up to 50% of those tests, at least in the genetic space, are going to be wrong? Because that's what the studies show. In fact, we're pulling the literature together right now. So it's interesting. So it's very attractive. It's incredibly lucrative. And I think it does have its place. But, it's tough. It's a big market right now. Direct-to-consumer is definitely where it's at. Our competitors are doing that, right? And it's great, right? You do a direct-to-consumer test, and you get an abnormal result. So you take it to your doctor. What does your doctor do? They repeat the test, right? Probably from the same lab maybe that did the test, right? So the lab's making it twice, and so that kind of grates my soul, so to speak. So but that's what I'm seeing is, it's not just kind of regular patient space, it's really wellness, and there are huge amounts of innovation right now.
well, if you're gonna make me ask the question, I'm gonna ask the question, which is, I have the benefit and the pleasure of working with Matt on some of his innovations out of the CLO and out of the university here. And this is gonna be kind of probably the final question I ask, which you can't have a panel about science ever without raising AI, right? And so in this prognostic space, there is a lot of retrospective data that we're able to now mine through and start putting pieces of the puzzle together in order to start identifying trends. And so going into that predictive space, going into that AI space, kind of the same-ish question I asked Tracy, which is, as an innovator, how do you see implementing AI and all of that data that's out there to maybe change the tune of prospective tests are only 50% accurate, to increase that accuracy and where you've been able to use that information on your own?
Yeah, so obviously an exciting time to be working in the space, and I do think that we're at the intersection where we're seeing some major advances in our ability to measure molecules, RNAs, proteins, cell-free DNA, circulating tumor DNA at unparalleled depth and at unparalleled scale. And as we do that, we encounter much larger data sets that we now have to interpret. But those are now being generated at a time where we do have breakthroughs in computation toolkits. These include obviously a number of large language models, artificial intelligence methods, and even quantum computing, where I think these methods allow us to more readily and more quickly separate signal from noise, identify patterns in these large data sets. I think those patterns, when these approaches are applied correctly, can be more accurate, which means they're more likely to be transported, they're more likely to be platform independent. And I think the promise there is that as we start to apply these tools to these types of data sets, the promise is that the signals we start to see may actually be more likely to represent true signals, not the false signals, and may allow us to actually develop these into clinically efficacious tests that really impact patient care more quickly and at reduced costs.
So Tracy, kind of same-ish again question to you is ARUP utilizing AI? If so, how are they using it? And what are you seeing coming out of current technologies that you're viewing or that you're generating in the space of using AI to hopefully decrease these false positives and increase accuracy?
So yes, I have an AI team that reports up through me, although I am not an expert in AI myself. We are doing AI specifically for applied AI. So that means we are specifically trying to solve problems in diagnostic medicine and pathology. So very specific problems in the lab, problems with pathology. So we can use, for example, there's a machine learning technique that we're using called the stool tool. We partnered with TechSite, which is a startup, out of Utah, to help them develop a really fabulous test, and it is better than a person, right, for detecting infectious organisms in stool. And now we offer the whole complete testing it's called ova and parasites, in our laboratory. It's done faster, honestly cheaper, and better using, image analysis, machine learning as a segment of AI. And we're looking at doing other types of testing with AI. But we decided to take a very narrow approach in terms of how we're going to use AI. We're looking at AI in terms of business and improving efficiencies, but I think that there's other players in that space, large players, that we're going to offer probably off-the-shelf solutions that we're going to incorporate and make our businesses even better and more productive. But I'd love to. We've got another product that's I think it's out of Hologics that looks at pap smears for cytology. Right? I don't know about you, but it's like spending 8 hours in a microscope looking at pap smears. Okay? Yes, I have done that as a pathologist. Far better to have image analysis, a machine learning tool that you can teach and learns better and can help identify the negatives from the positives. So then as a pathologist, this makes me way more productive, right? Because I know all these are the true negatives, and these are the might be positives, maybe positives. So I can concentrate my workflow on that. It makes me a much more productive pathologist. There's not enough of us to go around. So that's a nice example too.
All right, well, I promise to keep it short to allow everyone opportunities to speak with the other panelists here. So at this point, I'm getting to wind it up, Scott. So if I can kind of just wind it up, I'll say, again, I'll echo what I started with, which is you have an environment, you have an expertise, you have colleagues, you have resources here in Salt Lake City, on campus, outside of campus. Take advantage of it. Just by showing up here in and of itself, it's just a great opportunity. I work with about 50 different universities in what I do. Very few of them do this. Very few of them put on panels as good as mine. No, I'm kidding. But they don't put smart people in the same room, right? They kind of require those smart people to kind of find their way around. So take advantage of it. You're really blessed to be in an ecosystem and environment like this. So with that, I'll thank Tracy, I'll thank Matt, I'll thank the TLO and the other sponsors for allowing me to moderate this panel. And I wish you all a good evening. Please take the next hour, grab a soda or a cocktail, whatever is your preference, and mingle. And we're happy to answer any questions on the side. And with that, I bid you adieu.